Hi, everyone, and welcome back to Allied Health 209 Laboratory Testing. And so we've reached Chapter 8, Microbiology. And remember, your last two chapters on microbiology and toxicology, there will not be an individual test for those two chapters as we've had on all the other chapters. Just like the chapters you guys are, as I'm recording this, you will be finishing up reading chapter 6 and 7 on chemistry and immunology in your textbook. You will be watching the PowerPoint presentations for those two chapters in lieu of having your next test, which will be on chapter 6 and 7. But we will not have an individual test for chapter 8, microbiology, or chapter 9, toxicology. Instead, you will be responsible for the material in these two chapters, so you need to read your textbook, chapters 8 and 9. You need to watch the PowerPoint presentations, fill out your review guides, and then study the review guides that you have for your lecture final. Because there will be some questions from microbiology and toxicology on your lecture final, which is comprehensive. So let's get started. We can go over this material. We go over it a little fast, but you guys are able to rewind and go back anything that you missed. Plus, if you have any questions, comments, or concerns about chapter 8, 9, or any of the other chapters, please feel free to text me, call me, or reach out to me by email. So as always, you guys know I always like to start by talking about your learning objectives. And like I said, these are always important because anytime you're doing anything, you always want to have clear goals and objectives. And I want you guys to know what I expect you guys to have learned when you finish reading chapter 8 in your textbook and watching these PowerPoint presentations, and of course studying that material. You're going to be able to describe classifications of microorganisms, go through all the rest of these, all the way down to listing some of the emerging infectious diseases that we see. So make sure that you're familiar with these learning objectives. Make sure you understand this is what you are expected to learn during this process. And they're also listed in front of your textbook. And guys, really remember that textbook goes hand in hand with these PowerPoint presentations. And if you want to be successful, it's really a good idea to read those chapters. They have a lot of great information. There's a lot of great illustrations and charts in there that can really help you. So we always start with some review questions, right, from the previous chapters or from previous information. So I want to go through these because I feel like, if you don't want to use them, I understand, but they're really good tools for you to use during these times of this being an online class. If you can use these review slides as a way to test your studying. So you study for chapter 7, and then you can use these review questions as a way to see if what you're studying is locking into your mind. So you guys can pause each one of these and then see if you're getting the correct answer. I'm going to go through them rather quickly, but you can pause them. I'll give you enough time to pause as each new question comes up. Okay. So again, guys, like I said, I really put those slides up there as a way to help you because I know a lot of people do really fine on online classes, but some of you need a little help, those little extra things like flashcards and things of that nature to really help you study to see if that studying of those review guides are clicking in with you. So let's get started on our next chapter, okay? So microbiology chapter 8. So let's start with the fundamentals, all right? What is a microorganism? So we know that a microorganism is a tiny microscopic, can only be seen under the microscope, right? Entity capable of carrying on living processes. And what is microbiology? We understand that microbiology is the study of these microorganisms. And some of these microorganisms are things like bacteria, fungi, protozoa, and viruses. Okay, guys, now that we've defined what a microorganism is, now that we've defined what microbiology is, let's go into talking about what we refer to as normal flora, or sometimes it can be referred to as a normal biota. Now, I know y'all have heard of this, probably on television, your gut biome and things of that nature. Let's talk about this so we can understand it. The first thing I want us to understand is that our body has bacteria, normal flora in our body, that if in the correct part of our body, it does a good job, right? Not only does it do things to help us, but it also prevents other bacteria that can turn into pathogens from taking over, okay? So one of the ways that the bacteria that is present in a biofilm communicate with each other, okay? And the way they communicate is called quorum sensing. And this quorum sensing allows the whole biofilm to work together as one, okay? And so in order for bacteria in a biofilm to use that quorum sensing in a way that is continually active and responsive, they must possess three distinct characteristics. First, they must secrete a signaling molecule called an autoinducer, okay? Then they must be able to detect the change in concentration of signaling molecules. And then they must regulate gene transcription as a response. Now, to give you an example of this, lactobacillus is a normal bacteria in the female vagina. Now, I know a lot of you may have heard of this, especially a lot of women, that this lactobacillus, it creates a very acidic environment, okay? Now, that's all fine and good. That's what we need. That's what we want. That's an example of that biofilm causing that acidic environment inside the vagina that keeps it at where it needs to be. But we also know that this happens not just in females, but sometimes in children. We see thrush that forms in the top of their mouth. If you get on an antibiotic and you've been on that antibiotic for a little while, the side effect of that antibiotic can be not only does it kill the bacteria it was meant to kill, but it can also kill the lactobacillus, okay? The problem with that is we want that lactobacillus to be there, because once that lactobacillus gets killed or the numbers lower enough, it gives Candida albicans, which is yeast, which is an opportunistic infection. It gets that opportunity to move in there and take over, and then you end up with a yeast infection. We see it with children where that thrush, which is yeast, ends up in the top of their mouth, and it's because some of the normal flora that was in your mouth was killed off by the antibiotic you were taking and left a void. So the yeast or Candida albicans filled that void, okay? So there's a nice little cute video right down here that really goes into explaining the microbiome. I really like you guys to copy down this link. It's a YouTube video. It's really short. It's very cute, and it really, really helps you understand in a very entertaining way kind of how that microbiome works. So let's talk about pathogens. What are pathogens? Pathogens are organisms that can cause disease, right? Only a small number of organisms, less than 1%, are pathogenic for human beings. A person's immunity, which we just talked about in chapter 7, right, plays a large role in whether the pathogen will be able to produce a disease. It's like I talked about in chapter 6 when you have a person who is HIV positive. The AIDS virus beats down their immune system so low that diseases that most of us never get because our immune system fights them off, they get these very strange or opportunistic infections that other people don't get because their immune system is so suppressed. Healthcare workers must apply aseptic technique to prevent the spread of pathogens. This includes things like we talk about every day, right? Washing hands often, and especially between patients, wearing gloves, okay? And making sure that you change your gloves between patients, and especially wearing gloves in the appropriate PPE, not only with dealing with patients, but with dealing with any type of blood or body fluids. Okay, now we're going to move on and talk about some classifications of microorganisms. Okay, although complex identification procedures are not normally performed by medical assistants or lab assistants, it's important to have just a general knowledge of them. Okay, we always want to be educated in the field that surrounds us and to make sure that we're knowledgeable of what's going on with our patients, and so that we can always understand and communicate intelligently with doctors, patients, and other co-workers. So what are some examples of microorganisms? We have viruses, we have bacteria, fungi, or parasites. So let's start with the virus. What is a virus? It's a microorganism that consists of either RNA or DNA. They need to live in a host cell and use the host cell's organelles to multiply. They have to be grown on tissue cells. So what about bacteria? I know you guys learned a lot of this in early biology classes. We have prokaryotes, okay? They're classified as either prokaryotes because they have a cell wall and a cell membrane, but no true nucleus, as opposed to cells that are eukaryote, which have no cell walls, but do have a cell membrane and a nucleus with a membrane. So remember, study tip, eukaryote are like you. They have a nucleus. Okay, so we're still talking about bacteria, right? We're still listing out these things that define bacteria, right? They're usually single-cell organisms. They were produced by binary fusion, asexual reproduction that involved splitting that cell in half. They reproduce very rapidly. They form colonies of millions of the same bacteria when they multiply on media, as we see that picture down there in that far bottom right-hand side that contains nutrients for their growth, right? So we have nutrients in that media that's in that petri dish plate, and then we place a swab or some kind of sample of blood or body fluids onto that media. It gives that bacteria all the nutrients they need to grow so that we can identify which bacteria this could be, right? So now let's talk about fungi. Okay, we're going to list out some things about it, right? You see all these pictures of it under the microscope and on media over here to your far right. They also are grown on media, right? They're eukaryotes. They possess a true nucleus with a nuclear membrane. They lack chlorophyll. They are classified as molds or yeast. And remember, some fungi can take on both forms, the mold form being expressed at room temperature and the yeast form at body temperature. Yeast are single cells that reproduce by budding. And you'll see that picture right there at that top over to the far right, that little bud that comes out to the top. As you'll see it referred to as budding yeast, right? That's how they reproduce, in which a new cell forms on the surface of the yeast, grows, pinches off, and becomes a separate cell. They also form colonies, visible masses on the culture media that you'll see over also on some of the pictures over to the right hand side, and they have a really smooth, creamy appearance. If you notice the yeast growing on the culture media in the center picture over to the right, you can tell it kind of has that really smooth, creamy appearance. Okay, that's yeast, right? And then down at the bottom, we're looking at molds. Molds are made up of mycelium, which consists of tube-like structures called hyphae and have a fuzzy or woolly appearance on the media. And you'll notice that down in that bottom picture to the far right, that looks more fuzzy and woolly, right? Compared to that middle picture, right? In the middle over to the right, which looks very smooth and creamy. So here's one of our case studies, right? This is one of those things where you're taking the stuff you learn in lab and lecture and putting it together to see if you can answer those questions out in the field. So take the time to pause these, read through them, and see if you're getting the correct answer. Okay, now let's talk about parasites, okay? They can be one-celled protozoa, right, such as an amoeba, or many-celled organisms such as Hilmas, which are roundworms, tapeworms, and flukes. So you see that Giardia lambda, which is a protozoa up in that top picture over to your right. Then you have the hookworm in the center, right? Parasitic organisms live in or on a host and derive nourishment from that host. Carvin Lynn, a Swedish botanist, created the nomenclature system for all living things. Under the nomenclature system, how are the names of microorganisms written? So in the system, the genus is capitalized and the species is not. The genus and species are always underlined or italicized. The first initial of the genus, capitalized, may be followed by the whole species name in lowercase, such as S. aureus, or S. aureus underlined, or written out such as Staphylococcus aureus or Staphylococcus aureus. So let's go through this. What is the correct way to write Escherichia coli? So pause this and see if you can write it out the correct way. Okay, now let's talk about structural categories for bacteria, okay? So what basic shapes do bacteria take when they replicate, okay? Over there you see pictured over to the far right-hand side, we see this spirilla that looks like little spirals. You see the bacilli that looks like little rods, and you see the cocci that look like little circles, right? And that's pretty much what they are. And when you look at them under the microscope, depending on the particular bacteria, they will take on these shapes of spirals, rods, spheres, coccobacilli, which is a combination of both the cocci and the bacilli, okay? Okay, guys, make sure you know what each one of these shapes is talking about, including the coccobacilli, which is that cross between a spear and a rod. So now we're going to talk about the arrangement, okay? Obviously, the one is the coccus, right? The one spear-like piece of bacteria we see right there, right? And then obviously, Diplococci is a pair, right? Staphylococca is grape-like clusters. Streptococcus is chains, okay? Make sure you know down to the patterns, okay? That packs and patterns of 4, okay? You want to understand the arrangements that you may see these bacteria take when you look at them under the microscope. So now we're talking about the most common pathogenic Staphylococcus, and we want to give examples of diseases it causes, okay? The most common pathogenic Staphylococcus is Staphylococcus aureus. It is a gram-positive cocci, and it forms in grape-like clusters. It causes boils, it causes impetigo, abscesses, and wound infections. A lot of time, you will hear about it either like staph infection from a wound, you cut yourself and you get a staph infection, or we hear about it in food poisoning cases, okay? What about streptococca? What is the most common pathogenic, and what disease does this cause? So most is the pyrogens. It is a gram-positive cocci in chains, right? That's the most common pathogenic streptococcus, okay? It causes things like strep throat, scarlet fever, rheumatic fever, infections after childbirth, lots of others. So make sure that you understand, okay, that these are the most common of the strep, right? And we want to know which types of diseases or examples of diseases that this particular pathogen causes. So talking about the arrangement, right? What arrangement do the bacilli, now we talked about the cocci, what arrangement do the bacilli like to take? Same thing, right? Diplo, two diplobacilli arranges in pairs, and you see the little example of it over there to the right, okay? What are some of the diseases that bacilli causes? We see it a lot in urinary tract infections, botulism, tetanus, food poisoning, all the way down to TB, leprosy, and the plague. Okay, these are all different examples of diseases that this particular bacteria has caused. What about spirilla? Okay, gently curved or corkscrew or spring, right? And what are some of the diseases? We're talking about things like syphilis and cholera. So besides shapes, what are 3 other structures bacteria can have? We would talk about flagella, which is projections that resemble whips. So you see that picture up at the top to the right. I'm going to give you some examples in these pictures over to the right-hand side. There's a lot of really great examples in your textbook as well. Capsules, right, that surround the cell wall and make bacteria very vigilant because antibiotics have difficulty getting through that capsulated wall and getting in there and destroying that particular bacteria. Then we have spores, which are real resistant to heat or cold and chemicals in certain environmental conditions, which make them able to survive for many years. Bacteria will convert back to a vegetative infectious stage when conditions improve. And in other words, they may go into this dormant-like stage, okay, if conditions make it difficult for them to thrive. But the minute they get their conditions improved where they can survive, bacteria will convert back to that infectious state. That's why you hate to see people put on antibiotics and them not complete the regimen of antibiotics as prescribed completely. Because if you don't and you say, oh, I'm feeling better, I'm going to stop taking my antibiotics, the problem is you haven't completely done away with that bacteria, and after a little while you may have a resurgence, and it could even be worse than before of that particular bacteria because you didn't kill it off all the way, and now it makes that resurgence. So now we're going to talk about collecting, transporting, and processing microspecimens. So what are some safety precautions that should be taken when collecting microbiology specimens? This is something that's very important to medical assistants and lab assistants, as you very well could be the person responsible for collecting them, transporting, and processing them. First and foremost, as we talk about in lab, we want to wear the appropriate personal protective equipment depending on the particular pathogen we are dealing with. We want to make sure we disinfect counters after collection. We want to put specimens in appropriate transport bags after collection. And as always, we want to be diligent in washing hands after dealing with any patients or any specimens. So this is one of our thinking slides. So like I said, you guys, use these slides as a way to test to see if you're obtaining the knowledge you need. Because of course, these will be questions that will be on your test, okay, and on your lecture final. So pause these and see if you're getting the right answer. Okay, guys, you always want to make sure that you take every precaution to protect you, your patients, families, and your co-workers when you're working, okay? So continuing on this collecting, transporting, and processing, how should microbiology specimens be collected, okay? They're collected in a multitude of ways depending on the specimen, right? You want to make sure you collect before antibiotics are given and processed as soon as possible. Now, sometimes when we're talking about blood cultures, unfortunately, they have given antibiotics in a lot of these times before we can. But if the patient comes in and they have something new and we haven't done anything yet, you want to go ahead and try to collect that specimen before the antibiotic is given so we can make sure we know exactly what was in there. You always want to apply a swab from transport system, which is a culturette, and we've talked about these in lab before, and there's a picture of one in your top right hand corner. You'll notice there's that swab that you can swab the area, but then there's a place for you to place that swab down into, and a cap. You'll see the little white clear plastic cap that you place over the top. So there's media, there's stuff inside that transport tube to keep whatever you swab alive until they can plate it on the media. There's also an area for you to label your specimen appropriately. And as always, we make sure we positively ID our patient before we collect any sample. Once it's collected and put into the containers, things like urine, blood cultures, body fluids, skin scrapings, all of these things are things that you will collect and then put them into the appropriate collection container. We know that blood cultures can be drawn in multiple different sizes and shapes of blood culture bottles. Some even have chemicals inside them like resin that if your patient has already been placed on antibiotics, you want to draw the blood culture with the resin in there because it takes the bacteria, it takes the antibiotic out of the equation so that it makes it inactive so that the bacteria then can grow so we can find out what bacteria is in there, right? We always make sure we collect our urine in the appropriate urine container, and then we always want to make sure we collect the appropriate swab depending on where we're swabbing the patient at. We also have throat specimens that we have to collect, right? When we suspect a patient has maybe strep throat or some kind of bacterial infection of the throat, you always want to get your patient, and we went over this in lab, to tilt their head back, right? You take a tongue depressor to hold down their tongue. It's good to get them to say ah, and then you take your swab, stick it to the back to one side, and you want to rotate the swab in a circular or figure 8 pattern, avoiding teeth, tongue, or any area other than the throat. So pause this and see if you're able to get the right answer. Remember, we don't want it to touch the teeth and the tongue because we know we have normal bacteria all in our mouth, right? And we want to make sure we're getting that bacteria that we see in the back of the throat, maybe on those white patchy areas. We don't want to contaminate that bacteria with just some other bacteria that's on your teeth, tongue, or cheeks. So you want to be very careful to avoid those and go straight to the back and swab over. Now, documentation is always critical, right? We always want to double and triple check ourselves to make sure that we're charting appropriately. So how would you document collection of a throat specimen? Date and time always, right? And then you always want to say according to office policy and procedures, right? And then your initials and credentials. So still talking in this frame about collecting, transporting, and processing, right? When and how are blood cultures collected? Now, when a patient comes in with a fever that is of an unknown origin, okay, that is usually when doctors will order blood cultures. The most important part of blood cultures is aseptic preparation of the puncture site, right? And to make sure you keep good aseptic technique during the entire time you're collecting your blood cultures. Most blood culture bottles require approximately 10 milliliters of blood each for the aerobic and anaerobic bottles, and 1 to 3 milliliters for one aerobic bottle for a pediatric patient. The anaerobic bottle is filled first unless the butterfly method is used because, of course, we don't want to bring air into that bottle. In which case, the aerobic bottle is filled first because it's not going to hurt it to bring the air into it that is in the tubing of the butterfly setup. Here's one of those concepts, right, that I want you guys, because it is very important as healthcare professionals, as medical assistants and lab assistants, that you're able to describe these things in your own words. So describe in your own words why it is so important for blood cultures to be collected aseptically. Now take what you have learned in both lab and lecture and be able to answer this in your own words. Make sure your answer contains that collecting the blood aseptically means collecting it without any bacterial contamination at all. It is so important because you do not want the doctor to think the patient has septicemia, an often life-threatening condition, unless they truly do. So now let's talk about urine collection. We've went over this in lab, and we've talked about we've been collected some urine specimens. So remember a clean catch, right? A clean catch urine is when you clean your genital area with 3 soapy wipes. Then you start to urinate the first part of your urine into the toilet. You move your urine collection device into the urine stream, catch the midstream of the urine, move the container out, and urinate the rest of the urine into a toilet. So now let's talk about chlamydia. What is chlamydia and how is a specimen collected? This is the most commonly sexually transmitted disease, and it's caused by Chlamydia trichomonas, a tiny gram-negative intracellular bacterium that requires a host for cell growth, okay? It may have no symptoms in men, and in women, it can cause them to be infertile. I showed you guys a video of trichomonas in lab, and we talked about this. This is, like I said, the most commonly sexually transmitted disease, so it is something, as a medical assistant, you could very well come into contact quite a bit. So how do we collect a specimen for that? Physicians collect swabs from the endocervical canal of women or the urethra of a male and places it in transport media. You'll see that picture down the bottom right-hand side. That little container they're holding in their hand, they stick the swab where they've swabbed these areas down into it, and there's nutrients and media in that tube to keep that bacteria alive until we can plate it. Then you break the swab off at the end and then place the cap on the top of it to keep anything out of it and then label it properly. Another way of identifying this organism is a DNA probe test. So now let's talk about another sexually transmitted disease, gonorrhea. How is that specimen collected? We want to talk about the collections because that's what we're talking about, right? Collecting, transporting, and processing these micro specimens. Gonorrhea, which, like I said, is a sexually transmitted disease of the genitourinary tract caused by Neisseria gonorrhea, a gram-negative intercellular diplococcus, right? It's 2 kidney bean-shaped cocci somewhat facing one another. You see that in the top picture over there to your far right-hand side. Women will have no symptoms or dysuria and a yellow discharge. Men may have dysuria and a thick, creamy, whitish discharge from the penis. If untreated, this can cause infertility in both men and women. Physicians collect swabs from the endocervical canal of women or the urethra of a male. The specimen is swabbed on a Gembeck plate, okay? That Gembeck plate has a small white peel. The peel, when activated, creates an atmosphere of 10% carbon dioxide, right? Which is needed for the growth of Neisseria gonorrhea. The Gembeck plate, also called the Gembeck transport media, is then placed in a plastic bag. One in 4 people will have a sexually transmitted disease before they die. That's taken from the CDC. Guys, one of the things we need to do as healthcare professionals, especially if you're a medical assistant, is to make sure that we don't want people to feel embarrassed or ashamed of these sexually transmitted diseases. Because when they are ashamed and when people make them feel ashamed of them, they have less tendency to go to the physician when they start having symptoms. And as I've talked to you about in this presentation, it can lead to infertility. I would hate to see a young male or female get a sexually transmitted disease and be so ashamed to go to the doctor. They let it go on so long that they ended up having other medical complications, including not being able to have children. So we want to take that that stigma away so that when people feel like they have contracted a sexually transmitted disease, they feel comfortable to come into their physician and talk to their doctor or their nurse or their PA or their medical assistant about what's going on with them, and we make them feel like it's okay so they can get the help that they need. It also helps cut down on transmission, because the sooner people go in and get treated for these sexually transmitted diseases, we can decrease the transmission to more and more people. So how is Neisseria transported? Remember, we talked about this. In that Gembeck plate that supplies 10% carbon dioxide, which is what Neisseria needs to grow. Remember the kidney bean? Gonorrhea, right? So here's another one of our case studies. So you guys pause this, read through it, and see if you're getting the correct answers. We always want to make sure we document. Okay, now let's talk about OMP, ova and parasite collection, okay? Ova and parasite collection kits, okay, consist of 2 containers with preservatives, one with formalin and one with polyvinyl alcohol, okay? The containers must be filled to the appropriate line, and it will tell you on that kit with a feces specimen. And as I tell you in lab all the time, guys, make sure you follow the manufacturer's policy and procedure for those particular kits. Always read the directions and make sure you're following the directions to a T, okay? To transport specimens by mail to a reference lab, the specimen is placed in a container, which is then placed inside a mailing container, and a biohazard emblem must be affixed to every container, including the outside one. So in other words, when you place that specimen in a container, okay, the outside of that container needs to have a biohazard symbol on it. It needs to be labeled appropriately. Then it is then placed inside a mailing container, and that container must have a biohazard emblem on it. And then if whatever the outside container is that's mailed, every one of them must have that biohazard emblem on them. So after microbiology specimens are collected, the next step is to identify the infectious microorganism microscopically. So now we've moved on to collection and transport and processing of these specimens, right? Because that is a vital step in this. We must make sure that we collect these specimens correctly in the correct containers. We must make sure that we transport them in the appropriate time, in the appropriate container, to the appropriate department, right? And we must make sure that we process them appropriately. If they need a certain type of media, if they need a certain type of environment, we want to make sure we provide that to keep that virus or bacteria or parasite alive until we can look at it under the microscope, or until we can put it on media and grow it out so that we can do our cultures and our sensitivities in some cases, right? So now we're going to move on to talking about that microscopic end of it. We're going to talk about microbiology smears, stains, and wet mounts. So we're going to talk about how bacterial smears are prepared. So a portion of the specimen is generally smeared onto a slide or mixed into a solution with water or saline on a slide. The specimen is then heat fixed to the slide by passing the slide over a flame of a Bunsen burner or some other form of heat. Smears can also be fixed to the slide with methanol. Heat fixing denatures the protein and causes the swabbed material to stick to the slide. Then it is stained with a gram stain to pick up that particular bacteria's staining characteristics. Now remember, if we don't fix that specimen to the slide before we stain it, the stain will just wash the specimen away, and when you put it under the microscope to go look at it, you'll see nothing, right? So we must make sure we either heat fix it or it needs to be fixed with some type of methanol, okay? So Dr. Hans Christian Graham developed the Gram stain more than 100 years ago. The purpose of the Gram stain is to distinguish between Gram-positive and Gram-negative organisms. Gram-positive organisms appear blue and Gram-negative ones appear red, okay? And you see that in that picture up there in the top. So you put both stains on there and then they take on the stain depending on whether they're Gram-negative or Gram-positive. So what is the role of an MA or LA in preparing a Gram stain? Although medical assistants and lab assistants cannot identify a specific bacteria from a Gram stain, they should be able to prepare a stain slide, identify the bacteria as Gram-positive or Gram-negative, and give the shape and the arrangement. Okay, so the specimen for the smear is obtained from what 3 possible sources? So where can we get it from, right? The first one is obviously a direct smear, right? You just take the swab that you had swabbed from whatever site you collected from, and you make sure that all sides of that swab touch the slide, and you smear it onto the slide. Roll it directly on there, right? You can remove a colony from the Petri dish and rub it into a drop of saline or water, and then put it onto the slide. So that's coming, and you see that middle picture you see there, you see them with the little loop taking one colony that has grown onto that Petri dish with the media, and then they take that one colony, put it in some saline or water, make a little mixture, and then drop that mixture onto the slide. Or you can use a sterile swab dipped into broth containing the bacteria and then roll it onto the slide. So those are 3 different ways that we can obtain the specimen and make a smear, okay? So here's one of our case studies. You got to pause this, read through it, and see if you're getting the correct answer. Okay, now we're going to talk about the basics of Gram staining procedures, right? So crystal violet is purple. That's all bacteria is purple. Iodine is the trapping agent, okay? Then all bacteria is purple, okay? Then alcohol acidine, decolonizer, okay? Once you do that, then gram positive is purple and gram negative is colorless. Then you add that saffron red, and then you have gram positive is purple and gram negative is red. That's just that basic principle of Gram staining, okay? Where, like I said, the crystal violet is placed on there, and it just makes all the bacteria purple, right? I want to make sure y'all understand this. And then we put the iodine, and you see the little pictures down here at the bottom, and then that's the trapping agent, and then all bacteria is still purple, right? Then we put on that same slide, we wash it with alcohol or acetone, and that's what we refer to as a decolorizer, right? At that point, if that bacteria is gram positive, it's going to take on that purple appearance, and if it is a gram negative bacteria, it's going to become colorless. Then at the last, we add the saffron, which is the red, and then that's where we can get, as we look under the microscope, the distinguish between the gram positive, which will stain purple, and the gram negative, which will stain red. So from this staining procedure that we just talked about, what do you think the most crucial step is? The decolorization step, right? Because remember, we had everything's purple, right? Now we need to get to the point where when we put that saffron on, we can see the things that are red, okay? The decolorization step, if it's not decolorized enough, everything will be purple. If decolorized too much, everything will be red. What causes bacteria to take on the purple or red color from the gram stain? Okay, remember, gram positive but not gram negative organisms hold on to the crystal violet because of the larger peptoglycogen layer in their cell wall. Okay? Gram-negative organisms have a thick outer layer of lipoprotein and lipopolysaccharides, which are dissolved by the decolorizer. Okay? This allows the crystal violet to be washed away by the rinse cycle. Okay? So you can see where the gram-positive will hold on to that color, but the gram-negative can't because of the difference in the layers of their cell wall. Okay? So we understand that the gram-negative, because they have that lipoprotein and that lipopolysaccharide, that decolorizer is able to dissolve that, and then the crystal violet washes away in the rinse cycle, because we rinse between each cycle, right? So gram-negative organisms are therefore colorless, like we talked about earlier, after the decoloration and is applied, and then they turn redder after the saffron is applied. So this is why they're purple, right? With the first, they're purple, and then we don't get that decolorization until we put on that decolorizer, and this is why the gram-negative is decolorized and the gram-positive is not. So how do you perform quality control? Because I talk often, all the time, I know you get sick of me saying it, that you don't do any test on any patient until you do quality control, right? Well, what you do is you take a known gram-negative and a known gram-positive organism, and they're stained, along with the patient specimen, to make sure that the colors are correct. Because what you're doing is you're going to stain 3 slides: a known gram-negative, a known gram-positive, and your patient. So what should happen is if you're staining all 3 of them at the same time and you look at all 3 of them, then your gram-negative should be gram-negative, your gram-positive should be gram-positive, and that makes you feel better, or makes you know that that you did all 3 of the slides at the same time, so all of them should have came out correctly, which would make you trust what is on your patient, okay? So how are the test results reported out for a gram stain by the MA or the LA? You want to state the color as gram-positive, purple, or gram-negative, red, the shape and the arrangement, okay? So I want you to take the time to look at these organisms that are in picture A and B, and I want you to pause these and see if you can get the correct answer. So pause them now. So we remember that gram-positive takes on that purple color because it has that large peptoglycogen layer, right? And we understand that because the gram-negative bacteria has that thick lipoprotein and lipopolysaccharide layer, it is dissolved by the decolorizer, which then allows it to take up the red color. Here's one of our case studies, guys. Pause this one right here and see if you're getting the correct answer. Okay, guys, we're going to talk about acid-fast smears now, okay? So what are acid-fast smears used for? They're used for organisms in the Microbacterium genus. They're referred to as acid-fast bacilli. Bacteria appears red with acid-fast smears. So we're talking about TB, right? So one of them is TB, right? So what is tuberculosis? It's an infection in which tuberculous nodules are formed in the lung and is caused by Microbacterium tuberculosis, which is an acid-fast bacilli. And then there's a list of the symptoms, night sweats, pulmonary hemorrhage, on and on, right? So what is Microbacteria avium complex, MAC, right? It is an acid-fast bacilli that decimates throughout the body in patients with AIDS. Both TB and MAC have been on the increase as a result of patients with AIDS. We want to be able to name and describe two methods of performing an acid-fast stain. The Zeele-Nielsen method, okay? It uses heat, and the Kenyon cold stain mixes a detergent with the dye to break down the wax in the cell wall. Both methods use a carbofluorous solution as the primary stain. They decolorize with acid alcohol and counterstain with methylene blue. What are the two acid-fast bacteria that we talked about? Mycobacterium tuberculosis and Mycobacteria avium complex, MAC, right? So we talked about both TB and MAC. So what does the heat do in the Zeele-Nielsen method? It breaks down the wax in the bacteria cell wall. And what does the detergent do in the Kenyon cold stain? It breaks down the wax in the bacteria cell wall. A lot of times what you're seeing, I know you're seeing a pattern here, is a lot of times what we have to do is either break down or dissolve a cell wall so that a particular stain can get in and stain that particular bacteria. Do all acid-fast stains break down wax in the cell wall of the microbacterium? Yes. Okay, then let's talk about wet mounts now, okay? So what are wet mounts and why are they done? Wet mounts are done to view organisms in their living state to show their mobility. So we're going to talk about two examples of what wet mounts are used for. They're used to detect protozoa like Trichomonas vaginalis, which is a commonly sexually transmitted disease that produces itching and a frothy, creamy discharge. Trichomonas has a characteristic jerky movement. Now, I've showed y'all that video in lab before, but here's a link to the YouTube video that actually shows you that particular protozoa in its live movement motion. It's really good if you guys could copy this and paste this into your browser and watch that video. It's very interesting. Also, wet mounts are used to detect the presence of what we call clue cells, vaginal epithelial cells covered with Gardnerella vaginalis, which is a gram-negative coccobacilli found in women with bacterial vaginosis infections. They produce a grayish discharge and a fishy smell in the genital areas. There's another link to another video you guys can watch that is just very interesting for you guys to see. So I hope you take the time to copy these videos into your browser and take the time to watch these videos. They're very interesting, and I think they'll help you guys put things together. So here's one of our case studies. So take the time right now to pause this and read through it and see if you're getting the correct answers. Okay, now let's talk about the KOH prep. What is a potassium hydroxide prep? So a KOH prep is used to identify fungal infections, okay? These are infections of the skin or yeast infections with Candida albicans. The KOH, when mixed with the specimen on a slide and heated over a flame, is an alkaline solution that breaks down protein material such as skin, hair, or mucus, facilitating the identification of any fungus that may be present in the skin scraping or mucus. So what we're trying to do is break down all that other stuff so that we can see the fungus that may be there. And that's what that KOH prep is for. You're prepping it so that you can get rid of all that other stuff, affix it to that slide so that you can see any fungus that may be present so that you can identify it. So here's another one of our case study, guys. This is a real great way to see if you're taking all the things you're learning in lab and lecture, putting them together, and when you're out there working as a medical assistant, you're able to put this stuff together and really help your patient. So pause this one now and see if you're getting the correct answers. So let's talk about pinworm specimen collection, okay? So what is the genus and specimen name for pinworm? Entomebus vermicularis, okay? Entomebus vermicularis is the genus and specimen name for a pinworm. And then you see the little worm itself up at the top, and is the ova and parasite itself, pictures of the two different parts, okay? So what is the life cycle of a pinworm? Okay, so let's go through that first. So the female pinworm lays eggs in the anal area of the host, commonly a child. This process causes itching. The host scratches the area infecting the hands and fingernails with worm eggs. Then the host, like children do, put their hands in their own mouth or in the mouth sometimes of family members. The eggs are swallowed and mature in the intestinal tract. Again, guys, I'm trying to include these really interesting YouTube videos to give you some insight on what a lot of stuff looks like. So really take the time to copy and paste these links into your browser and watch these YouTube videos that give you a lot of insight into this stuff, okay? Along with all the great information that's included in your textbook. There's a lot of great pictures and illustrations of not just the pinworm itself, but the process for collecting the pinworm specimen and identification, along with the things that we are also that we will be going through in lab. As we have now moved the remainder of our labs online, these last few, I will have PowerPoint presentations up going through a lot of those labs we would have done on KOH. So we're going to talk about tests used for pinworms. Okay, and there's a lot of great info in your book on this too, okay? Because the pictures on these, I realize, are a little small, but the ones in your book are real good as well. So the cellulose tape test, right? It consists of laying cellulose tape, okay, sticky side up over the person's anal area and then placing the tape sticky side down on a glass slide. So as you can imagine, they put them on a wooden applicator stick, they put the tape on it, and then they touch it in near the anal area on both sides, hoping that the eggs stick to the tape. Then you lay that tape where the eggs would be on that sticky side, tape the sticky side side down on a glass slide. Then the slide is scanned on oral immersion to look for pinworm eggs, which appear oval with a flattened side. It's a picture we see down in that bottom right-hand line. Collections are usually made on 3 consecutive mornings before the child rises. The MA or LA collects a specimen and then prepares and focuses the slide. A physician or trained laboratory professional microscopically examines the tape on the slide to look for Entomebus vermicularis eggs. So here's another one of our case studies, okay? So pause this and see if you're able to detect these correctly, okay? Remember, documentation is vitally important. Okay, so now we're talking about CLIA wave microbiology test. Okay, the first one we're going to talk about is Streptococcus group A testing. So some streptococci are capable of producing hemolytic toxins which hemolyze RBCs when grown on sheep blood agar. What that means, guys, that top picture over to your right, you'll see the little red edge. But if you notice, anywhere where you can see that bacteria is growing on that agar plate, it's not red anymore, because the streptococci is able of producing this toxin that hemolyzes those blood cells, and so they're not red anymore, okay? And you'll see it on that middle one too, how it's red, but then up there in that area where you see they streak the bacteria on there, it's almost clear or white, okay? And then part of the identification of Streptococcus depends on which hemolytic reaction occurs on the blood agar. That's one of the tools that the microbiologist uses to identify which Streptococcus it is. So we can talk about the 3 types of hemolytic reactions that they're going to be looking for on those blood agar plates. There's alpha hemolysis, which is incomplete hemolysis of the RBC seen as a green color around the colonies, which you'll notice on our top picture to the right. Then we have beta hemolysis, which is complete hemolysis of the RBCs seen as a clear area around the colonies. That's our middle picture over to the right. Then we have gamma hemolysis. It occurs when no toxin is present and therefore no hemolysis is seen around the colony. So remember, alpha hemolysis, that top picture, is incomplete hemolysis, that green color. Beta hemolysis in the center is complete, where it's white around that colony. And gamma hemolysis is when this particular strand did not release toxins, so you see no hemolysis around the colony of bacteria. So now I want you to take the time to pause this now and see if you can detect which A, B, and C, which hemolysis they are. So pause it now. So Streptococcus group A testing. So what is the gram stain reaction, shape, and arrangement of the Streptococcus species? Because remember, you want to understand the gram stain reaction that occurs, the shape, and the arrangement of these species. They, Streptococcus species, are gram-positive cocci in chains. You'll see the little chains up there, right? What is the hemolytic reaction of Streptococcus pyogens when grown at a blood agar plate? They show beta hemolysis, right? That's that complete hemolysis where it is clear and white around the colony. So purple, right? Beta, purple, gram-positive cocci in chain, right? With beta hemolysis, which is complete hemolysis. Streptococcus species can also be divided into groups A to O according to the ability to induce the formation of specific antibodies in their cell wall. What group is Streptococcus pyogens? It's strep group A. So you need to understand that Streptococcus pyogens is group A. What very common disease is caused by Streptococcus pyogens and what are the symptoms? Strep throat, right? Strep throat is the common disease caused by Streptococcus pyogens. The symptoms are very sore throat, right? A bright red pharynx, white patches on the tonsils, swollen glands in the neck, fatigue, and muscle pain. We want to note that bacteria are responsible for only 20 to 30% of pediatric cases and 5 to 15% of the adult cases of sore throat. The majority of sore throats are caused by viruses. Viral infections are commonly have symptoms such as a cough, rhinorrhea, hoarseness, and oral ulcers. Look, I feel like I can't talk today. So, bacterial infections with group A strep commonly have symptoms such as sudden onset of pain, painful swelling, and fever. So now we're going to name and describe the 3 complications of an infection with the Streptococcus pyogens. There's scarlet fever, rheumatic fever, and glomerulonephritis, right? So scarlet fever is a contagious disease characterized by sore throat, fever, enlarged lymph nodes in the neck, flushing of the face, and a strawberry tongue and a bright red rash, okay? Those are that. It's very contagious. Then we have rheumatic fever, which is a symptomatic inflammatory disease that may cause the brain, heart, joints, or skin, or subcutaneous tissues, okay? Then we have glomerulonephritis, which is an inflammation of the glomerulus of the kidney characterized characterized by decreased urine production, edema, and protein in blood in the urine. Other complications of untreated or partially treated strep infection. Okay, these are them. All the way from itis media, all the way down to throat or tonsil abscess, and then there's things like toxic shock syndrome and meningitis. So in other words, this can be serious. Again, we always, if we feel sick, we need to get to the doctor and find out what is wrong with us. And we need to make sure we're very vigilant as healthcare workers to make sure we do everything properly, to make sure we properly diagnose, monitor, and treat our patients. As some of these things that we sometimes see as common really can turn into things much more serious. So here's one of our concepts where we want to be able to describe things in our own words. So I want you guys to be able to describe in your own words how strep throat is different from other throat infections and why it is clinically significant for strep to be identified. Make sure you mention that it is different because it is so dangerous if left untreated. It is important that strep be identified because there are so many dangerous complications. And even though we not know that really being exactly positive for strep is not as common as people think it is, it's usually viruses that cause that. We want to make sure if this person possibly has strep, we immediately identify it properly as strep and then treat it due to the dangers if we go untreated. Streptococcus group A testing, right? Okay. So how do we test for Streptococcus pyogenes? First, a rapid strep A test is run. If it is positive, then strep is present. A positive test is denoted by the presence of a blue line and a pink control line. Okay? A negative test is denoted by no blue test line and a pink control line. Remember, these tests you must follow the manufacturer guidelines. And there's pictures really good in your textbook. There's some up here at the top. Remember, these internal controls must work. If your internal control line does not come up, then that test kit is not good and must be repeated. Remember, every new test kit, when you open it, you run external controls. Once you run external controls on the new test packet, then each time after that, you just want to make sure that your internal control works. And you see our positive, the internal control comes up positive, and then the positive line comes up positive, okay? In the next one, this is negative. The reason we can call it negative is because first, the internal control worked. We don't call any test if the internal control doesn't work. It worked, but there was no blue line for the test. So that means it's negative. Now, this one to the far right shows you invalid because there's a negative one where no line showed up in the test, and there's a positive line that showed up in the test. But neither one of them did the internal control come up, so they are invalid. Okay? A negative test does not conclusively mean that they do not have strep A. If it's negative, it should be followed up with a culture with a bacterium disc A because the specimen may not have contained enough organism to be detected by the rapid strep test. Beta hemolysis and no growth around the bacterium disc is a positive test. So up at the top, we see examples of our rapid strep A test. You see the examples of positive, negative, and invalid. You see the test kit that it comes in. Okay, and then our bottom picture to the right is this culture that we cultured the swab that we took from their throat, and we dropped that disc on there. Okay. When we see that beta hemolysis, which is that clear around the colony, and there's no growth around that A disc, then we know that this person has strep, okay? So if we get a positive on our rapid strep test, then we move forward with treating this patient as they have strep. If we get a negative and we're not sure, because of the seriousness of strep, if they have all the signs and symptoms of strep, we really need to do this culture just to make sure. So preparing the culture consists of swabbing a blood agar culture plate with the throat specimen. The agar is then streaked for isolation, as you see in that top portion, and I talked about this in class. You swab the swab onto the blood agar plate. Then you take your loop and you take an amount of that that's in that swabbed area with the specimen, and you start pulling it out to try to isolate down to one colony. A disc impregnated with specified amount of the antibiotic bactericin, called a disc, is then placed on the swabbed area. The plate is incubated overnight at 37 degrees Celsius. After 24 hours, the plate is observed for Streptococcus pyogenes, which will cause both beta hemolysis on the blood agar plate and no growth around the strep A disc. The positive results of beta hemolysis and inhibited growth around the bacterium disc are seen on the left, and the negative results of no hemolysis and no inhibited growth around the bacterial disc are seen on the right. So you see the difference between a positive and a negative. So here's another one of our little collaborative activities, okay, where you read through this and see if you can figure it out. So we talk about Tabitha Jones presented to her doctor with a very sore red throat with white patches on the tonsils, swollen glands in the neck, fatigue, and muscle pain. She had fever of 102 degrees and felt nauseous. The doctor ordered a rapid strep test and a culture. How would you collect the culture for the rapid strep test and for the culture? Pause this and see if you get the right answer. One swab performed the rapid test. For the rapid test, another swab that you rub on the blood agar plate. If the rapid strep test was negative, would this mean conclusively that the patient did not have strep throat? And what follow-up test should be done? No, right? Even though that may be negative, there may not have been enough sample on the swab, so we need to take that second swab and do a culture and sensitivity with the A disc to make sure we totally rule out strep because of the seriousness of the complications if left untreated. So let's talk about influenza. What is the etiology of the flu? The flu is caused by the influenza virus that affects the respiratory tract. There are three types of influenza virus, A, B, and C. Type C is milder. The elderly and patients with chronic problems are at greater risk of complications from the flu. The symptoms of the flu are a fever 100 to 103 and higher in children, cough, sore throat, runny or stuffy nose, headache, muscle aches, and fatigue that can last from 1 to 2 weeks. Some individuals develop complications such as pneumonia. So, influenza viruses are capable of mutating. Okay, that's the problem with viruses as a whole most of the time. So what is the difference? We're going to discuss the difference from antigenic drift, antigenic shift, and a pandemic, right? An antigenic drift is the gradual change that these viruses undergo from one season to another, okay? Antigenic shift is an abrupt change, okay? And then antigenic shift causes large numbers of people to contact the disease, and then it's known as a pandemic. So like with COVID-19, when you heard it's a novel virus, that means it's a new virus that we haven't seen before. So then when it comes into the public, because our immune systems have never seen it before, that's why it causes what we've seen with this global pandemic. It's new. A lot of people get it. We don't have a defense to it. We know nothing about it. It's all new, right? Okay, pandemics are deadly. We know this. We've seen this with COVID-19. We've seen it earlier with the Spanish flu that caused 500 deaths in the United States, 20 million worldwide. The Asian flu caused 70,000 deaths in the U.S., and the Hong Kong flu caused 34,000 deaths in the United States. So what test is used for influenza? The qualitative lateral flow immunoassay test that detects influenza type A and B. And you see some examples of these test kits on the right-hand side. There's some really great pictures and details in your textbook. So what is the difference between an antigenic drift and an antigenic shift in a pandemic? So an antigenic drift is the gradual change viruses undergo from season to season, right? So we don't see a really big spike in anything. They just gradually go through the change, and they're going through it so slowly, we slowly adapt, right? We don't have an overload to the system. An antigenic shift is an abrupt change, okay? And so a pandemic is when large numbers of people contact that disease after an antigenic shift, or if you see a new and novel virus. So what are three groups bacteria can be broken down into on the basis of their need for oxygen? As I've told y'all before, we're going to give them all the requirements they need to grow because we want them to grow so we can find out what is going on inside your body. And that means if they need 10% carbon dioxide, if they need a different media, growth media, different nutrients in that media, different temperatures, different transport, we're going to provide them with that. Some things require oxygen for growth, aerobic organisms, and then we have anaerobic organisms that can grow and function in the absence of oxygen. And then there's facilitative anaerobe, an anaerobic organism that is also capable of growing in the absence of oxygen, okay? So growth requirements of bacteria. So we want to describe an example of an anaerobic organism, the disease it causes and how to prevent it. Clostridium tetani grows in deep wounds and produces deeply neurotoxins that cause tetanus and lockjaw. Patients with a puncture wound need to get a tetanus shot or injection. Here's some examples of what can happen to a person left untreated. So how are anaerobic conditions created for the growth of bacteria that grows in the absence of oxygen? We see an example of a setup over here to our right. A gas-generating envelope is placed in a container. 10 milliliters of water is added, and the container is sealed down and closed airtight. The water generates hydrogen and carbon dioxide, which reacts with oxygen in the container's air to create an anaerobic atmosphere. You'll see those plates are placed down into that, and then you set this container up like this so it produces that anaerobic environment. There's different types of growth media, and we're going to give an example of some. There's nutrient medias. They're made from meat and soybean extracts like soy agar. There's selective media, right? It inhibits some bacteria while allowing others to grow. There's the McConkey. It inhibits gram-positive organisms. Then we have what we refer to as differential media. These visually show metabolic differences between bacteria. We have McConkey shows lactose fermenters as red. So that's very helpful in us identifying certain types of bacteria. And then we have what we refer to as enriched media. These are media that contain enrichments, like blood agar contains sheep blood, or chocolate agar contains blood that has been heated. And there's an example, and you can see from the example the blood down at the bottom to your left, and then the chocolate, which is the heated blood, over to your right. But these nutrient, selective, differential, and enriched are all different types of growth media. So here, pause this and see if you get the right answers. So what do you call organisms that require specific additional additives and conditions for growth? Fastidious organisms, right? Fastidious organisms. So what is an example of a fastidious organism, and what additional conditions does it need to grow? So Neisseria gonorrhea is an example. It needs blood serum, amino acids, vitamins, and it needs that 10% carbon dioxide. So we want to identify from its description 5 large pieces of microbiology equipment. We have an incubator, and I'm giving you examples over here to your right. Incubator, it's used for growing cultures at body temperature. You've seen the one that we have in our lab. There's safety hoods. That's an area that if you're working, you get underneath that. It's an area with a fan that pulls the airflow away from the person sitting at the safety hood, so that any noxious things that you wouldn't want to breathe in go straight up that fan and out, right? Then you have what they refer to as an autoclave. It destroys microorganisms by applying steam under pressure. Okay, that's the way you can make sure that you kill everything. Then we have refrigerators, right? We need those for storage of culture plates and supplies, right? Some stuff needs to be kept at room temperature, some needs to be kept frozen, and some needs to be kept in a refrigerator. And we need to always read the directions so we make sure we're storing all of our stuff at the appropriate temperatures. Then we have microscopes, right? You use those in the lab. They're used to identify the microorganisms that we've been talking about. Let's talk about some other microbiology equipment. We want to be able to identify from its description small pieces of microbiology equipment. We're going on further with these. We're looking at stuff like loops and needles, Bunsen burners, and electric incinerators. So look at the top to the far right. These are loops and needles, right? These are inoculating loops that has a bubble wand at one end and comes in varying sizes, so a precise amount of specimen can be collected, such as a urine culture loops are calibrated to collect either 0.01 mLs or 0.001 mLs. So that loop is calibrated to that amount. So as that cooled, that loop is sterile. Okay, you sterilize it with your electric incinerator, and then you let it cool back down. Then you stick it into the urine and it collects that precise amount. Then you can take that loop and then take that urine that's inside of it and put that exact amount on a culture media. Then you have what we refer to as inoculating needles, pointed at the end and used for picking up colonies of bacterial growth. If we've seen on a lot of these culture media, these little round, some of them are smooth looking white, that's a colony of bacteria, and we can use these inoculating needles to pick that colony of bacteria up and put it in broth, media broth, to put it on another plate, whatever we need to do. Then there's Bunsen burners and electric incinerators. These are used to sterilize loops and needles before and after each use. So you stick, as you see, you know you would hold it over the Bunsen burner flame. The electric incinerator, you can stick the end that has your loop on it in there. It's going to sterilize that loop, then you take it out. And like I said, you need to wait till it comes back to temperature to be able to then stick it into urine and collect your urine specimen. The electric incinerator is much safer than the Bunsen burner because it does not use a flame to sterilize the loops and needles. Okay, so you work in a microbiology reference lab as a laboratory assistant. Work with your lab partner to write down as many pieces of equipment as you can be found in the lab where you work. Okay, what I would like you to do, since we're not in the lab room, is I would like you to take out a piece of paper right now and pause this and see if you can write down as many of those pieces of lab equipment that we've talked about today. This is just a way, guys, for you to work these things out, and it's a way to study while we're going through this, so that when you take your lab final, you'll get a lot of these things right. So this would be a really good activity for you. You can pause that now and complete that activity. So, inoculation of media, right? And then you'll see down there at the top picture on your right is someone collecting a throat specimen. The picture down there in your far right-hand side is someone taking that throat specimen and placing it on a blood agar plate so that we can allow whatever bacteria may be growing in our patient's throat to grow out here so we can identify. If the patient's symptoms indicate that a bacterial infection is present, the physician will order a culture and sensitivity to determine what bacteria are present, that's the culture part, and what antibiotics would be best to kill that particular bacteria, which is the sensitivity part. So we understand that a swab is used to collect the specimen, then it is introduced onto the media used to grow the bacteria. So after the specimen is collected, what are two ways it is plated or streaked onto a collection media? Like I said earlier, we talked about swab specimen. such as taking a throat swab and directly rolling it across the culture media, touching all sides in the upper part of the Petri dish. If the specimen is liquid, such as urine, then an inoculating loop is used. After sterilizing it, it is dipped in the liquid, then swabbed onto the top section. Okay, just like the picture in the far bottom right-hand side shows, you just inoculate that specimen on that top corner of that Petri dish. Then we have to spread the specimen onto the culture plate, right? We talk about quadrant technique or isolation streak. You take the sterilized loop and spread the specimen that you put in that top corner over 4 areas of the plate to establish isolated colonies of bacteria that then can be identified. And you see the streaking pattern up in the top. Okay, you start with the area that's the thickest that you put the swab or the urine itself onto, and then you take your inoculation loop, and you'll see that green in that top corner over to the far right. If you see the green, that's the first where we try to drag it out, right? We drag it out and try to isolate it, and then the blue, you try to isolate it a little more, and then the reddish color when you try to isolate it until you can get it down to there's that one round colony of bacteria. Better isolation is achieved if the streak lines are kept close together and the third and fourth quadrants do not overlap. Now that first at the top on the right hand side is showing you how you streak it. Bottom is showing you a real culture that is grown from the streaking. If you see it squiggle out to the end there, you see where we might can get that one isolated colony of whatever bacteria is growing in this patient. Colony count or long streaking, okay? This is another way. It involves streaking the entire plate. In the colony count method, the whole plate is streaked with a calibration loop so that the colonies that grow on the plate can be counted. Example is a urine culture, okay? In the long streak method, a swab that has been dipped in a broth containing only one kind of organism, which we refer to that as a pure culture, is swabbed on the entire plate. Okay, this is when we're doing antibiotic testing. You've already grown the bacteria out. You've already isolated that one bacteria. You grew that one bacteria in some kind of broth, and then you took that isolated colony out, long streaked it onto this plate, and then from there you can drop. You see that bottom one. The top one is the long streak method. It shows you just like before. Then we see the method where we got the colonies all grown out on there, right, in that middle one. And then down in the bottom, you see those little discs that have been placed out there. That's the sensitivity part. Each one of those discs has a different bacteria. And the one with the biggest area that killed the bacteria around them is the antibiotic that's going to work best on this particular organism. So here's another one of those sections, guys. I can't tell you how important it is for you to be able to describe in your own words. Then you would be able to speak intelligently about the things you're learning. So in your own words, you want to talk about the two main methods of streaking bacteria on a culture plate and why they are used. You want to make sure your answer talks about the quadrant technique or isolation streak, right? A specimen spread over 4 areas of a plate to establish isolated colonies of bacteria used to produce pure cultures of bacteria so they can be identified, right? Colony count or long streak method. Colony count is a whole plate of streak with a calibrated loop so the colonies can be counted to see if it's a urinary tract infection. Remember, we have a threshold. There needs to be, if it gets to X amount of colonies, then we say, yes, they have a urinary tract. If it's in a middle area, then we say that's a possible urinary tract infection. And if it's below that, then we say it is not a urinary tract infection. And remember, long streaking is a swab with one kind of organism from that pure culture is swabbed onto the entire plate. Then antibiotic discs are added to see which antibiotic the bacteria are sensitive to. As the specimens are streaked on the appropriate media plates, the plates are placed in an incubator. The temperature of the incubator is usually 37 degrees Celsius or body temperature. So how's a urine culture performed? The urine culture is collected by the clean catch method. We talked about that earlier. A colony count is performed using the Longstreet technique, a calibrated loop, usually one containing 0.001 ml of specimen, is used to cover the entire culture plate with the urine. After urine is incubated overnight, the colonies that form on the media are counted. And we see those colonies down in that far right-hand side media plate. The number of colonies counted is multiplied by 1,000. Fewer than 10,000 colonies per milliliter is normal. Between 10,000 to 100,000 colonies per milliliter is considered suspicious of a urinary tract infection. More than 100,000 colonies per milliliter is indicative of an infection. You guys need to know this information. The number of bacteria grown is more significant than the identification of the bacteria for the diagnosis of UTI. Pause these now and see if you get them correct. So what is one way bacteria are identified biochemically? So a pure culture, and we talked about how we got a pure culture, right? Consisting of only one kind of bacterium is mixed with an enzyme on the slide. A positive test shows bubbling. The catalyst test differentiates Streptococcus, which is negative for the enzyme catalase, with Staphylococcus, which is positive for the catalase. So we understand that this is a chemical analysis we can do that can help us positively identify the difference between a strep infection and a staph infection. So biochemical testing using methods such as microscan and intertube takes longer to establish results. And there's an example of your microscan and an intertube down at the bottom. Automated instruments can be used for the identification of bacteria. And here's some examples that Viatech and the AutoBac. The BacTech determines if bacteria is present in blood cultures. These are all automated instruments that you may run into in larger hospital or reference laboratories. So here's one of our little collaborative bacteria, right? So read through this one and then talk about how should the specimen be collected. So pause it and see if you're getting the correct answer. Remember, documentation is vitally important. If you don't document it properly, it didn't happen. So let's talk about sensitivity testing. Why is sensitivity testing performed? Physicians order sensitivity testing to determine what antibiotic to use to treat that bacterial infection. Guys, we know that there are a lot of side effects from any medicine we take sometimes, right? So we want to make sure that these bacterial infections don't proceed to something worse, right? We know we can get one and they'll turn into something else. We also don't want to take an antibiotic if it's not working, and we want to try to get rid of it as fast as we can. So these sensitivity testings can literally show us which antibiotic works the best against this particular organism. And now we want to talk about what criteria our physicians use when choosing antibiotics to prescribe to our patients. So which antibiotic showed no growth around its disc, meaning the bacteria was sensitive to the antibiotic? That's the first thing they're looking for. Which antibiotic is the least toxic to the patient and the patient's normal flora, right? And which antibiotic is the most cost effective? So remember, they may not go with just the one who killed the most antibiotic. We need to make sure that antibiotic is going to be the least toxic to our patient. And we talked about earlier how it can kill off the normal flora and then just cause another opportunistic infection, right? So we got to be careful of all that. And then we have to be cognitive of cost, right? Some of our patients are on fixed incomes, and we don't want to prescribe them a medicine that they can't afford because they're not going to take it at all. So it would be better to prescribe them one that may not work as well, but does work and is more cost effective for them to make sure they actually fill that prescription and take the medication, right? It's those little things sometimes that we need to take into consideration. That medicine is not going to do our patient any good at all if they can't afford to get it. So it might be better to use one that doesn't work quite as well, but works, that is cost effective enough that our patient can actually get it filled and be able to take it. We want to talk about two methods of performing sensitivity testing. We have the Kirby-Bauer method, where you inoculate a liquid broth with a pure culture of the bacteria to a specified turbidity. Spread the bacteria over the whole plate in that long method we talked about of appropriate media, right? With the Kirby-Bauer apparatus, okay, discs containing a specific amount of antibiotics are dropped onto the inoculated plate and incubated overnight. And there's a picture in that, the picture at the top is our pure culture growing in our broth, right? That middle picture is the Kirby-Bauer method. The little blue thing to the other side is one that you pick up, you can place it over that disc, the agar, and you push down in the center, and each one of those little clear things with the white disc in it will drop down a different antibiotic in perfect order, spaced out correctly, so that they work well, okay? It's a really nice system. So we incubate it overnight. Each disc is then checked for zone of inhibition or of no growth. The zones are measured in millimeters and compared with the manufacturer's values and are recorded as either sensitive, intermediate, or resistant. So, since it's still talking about sensitivity testing, the minimum inhibitory concentration, MIC, right, involves using plates with wells in them that contain a serial dilution of the antibiotic. A liquid is inoculated with a pure culture of bacteria to a specific turbidity. Equal amounts of the organism is added to each well. For each antibiotic, the first well of dilution that does not have any growth is the MIC, okay? This method informs the physician of the exact amount of antibiotic needed to inhibit the bacteria. This can be done both by manual method and with automated technology. Now, as you can imagine, the greatest benefit of this is to give our patients the least amount of medicine humanly possible, but giving them enough to make sure it does the job, right? So we don't give them more antibiotic than they need. We want to talk about the pathogenic bacteria and its description that commonly causes each one of these diseases. So strep throat, Streptococcus pyogenes, right? It's a gram-positive cocci in chains. Top picture over to your right. Urinary tract infections. More than one thing can commonly cause urinary tract infections. The most common are Escherichia coli, Protus, Klebsiella, Pseudomonas aeruginosa, all gram-negative bacilli. And then we have pneumonia. That's our middle picture, and then our bottom picture is pneumonia. Streptococcus pneumoniae. It's a gram-positive diplococci that looks like cat eyes. Okay, you see those in there. We have wound infections, which are Staphylococcus aureus, which are gram-positive cocci and grape-like clusters. That's your top picture to your right. Gonorrhea, which is Neisseria gonorrhea, which is a gram-negative diplococci that are shaped like kidney beans facing each other. That's your middle picture. And then chlamydia, which is a group of bacteria that can reproduce only in a cell. Okay, so there's an example over to your right of each one of these. And these are pathogenic organisms that we see quite frequently in physicians' office labs. Okay, still talking about pathogenic organisms that we see frequently in physicians' office labs, we're going to talk about pathogenic fungi. We're also going to, same thing, we're going to give descriptions that commonly cause each of these diseases. Vaginal yeast infections and thrush. That's Candida albicans. It's an oval shape up there at the top, over to your right. Organism that take up crystal violet and can be budding. In the middle, we have athlete's foot or jock itch, ringworm. All right, there's lots of different microspores showing hyphae or branching, right? And let's talk about some parasites. We have pinworm, right? Intermebus vermicularis is a worm identified by the flattened oval egg on the cellophane tape test. We have roundworm, right, identified by eggs in the patient's stool. We have lice, is a body louse, and it can be crabs in the pubis area, and both lay eggs and are called nits. Okay. Giardia lambda is a protozoa found in the stool specimen that causes abdominal discomfort such as cramping, diarrhea, and nausea. We have Trichomonas vaginalis, is that sexually transmitted protozoa that exhibits jerky movement and causes vaginal itching. Guys, you're going to want to know all this stuff, okay, for the test, okay? A lot of this will be on your lecture final. Plus, these are things that you are truly going to run into each and every day working out in the field. So, name a gram-positive cocci and chain that causes sore throat. I want you to pause each one of these and see if you're getting this correct. You're going to know these guys, so study these things, especially the Streptococcus pyogenes, Candida albicans, your Escherichia coli, and your Entamoebis vermicularis. So let's move on to talk about emerging infectious diseases. So the relationship between microorganisms and disease is not fixed. New infectious diseases may emerge suddenly. We all know this. We've seen it when it happened with COVID-19, whereas others seem to disappear and then reemerge. According to most experts, the rate of which infectious diseases emerge and reemerge has risen in recent years. Some of the most important reasons for this trend are the growth of our human population, the rise in international travel. We can spread disease from one area to the other very quickly, and environmental changes due to global warming. So we want to talk about 5 emerging infectious diseases and give their description. SARS virus was a precursor to what we talk about now with COVID-19. Okay, this is a respiratory infection caused by the coronavirus, right? Reported in Asia, North America, and Europe. Okay, we had the hantavirus, which was a highly fatal respiratory infection spread by deer mouse, isolated in the southwest U.S. We have HCV, which is hepatitis C virus. That's a liver disease that infects up to 3% of the world's population. Escherichia coli, or 157H7, causes hemolytic uremic syndrome, severe anemia, and kidney failure. And then we have, of course, the human immunodeficiency virus, HIV, which causes AIDS. Now we're going to talk about bioterrorism, okay? So in the aftermath of the bioterrorism threats following 9/11, all laboratories should perform a risk assessment. We should all have a risk assessment, not just for emerging public health threats such as viruses and bacteria that can cause epidemics and pandemics, but also bioterrorism issues that can happen. So laboratories must identify procedures and methods that have potential to produce aerosol, which is fine particles suspended in the air, right? Using biosafety cabinets or hoods to perform tasks in ways to avoid exposure to aerosols. The CDC recommends referring any suspicious organisms or substances to the proper authorities and practicing the proper biosafety techniques at all times. Most of the bacteria, viruses, and toxins used as bioterrorism agents can be spread by an aerosol route, which is very stable. They also produce high morbidity illness, which is illness, and mortality, which is death rates, right? Other bioterrorism agents are transmitted person to person, and some are difficult to treat. We want to talk about 6 of the most feared bioterrorism agents. Anthrax, or the wool sorter's disease, is caused by Bacilla anthracis, a gram-positive spear-ended shaped rod that forms endospores. Okay, there's a picture up there in the top right that shows you those spear-ended rods, right? It is disease contracted by livestock when they feed on plants contaminated with the organisms. So it goes to the lambs and to the cows and stuff first, right? Then humans contact the disease by coming in contact with the animals. like wool sorters that were sorting those wools taken from the lambs. It is not spread from person to person, but by contact with the spores themselves. It has three forms: cutaneous, which we show a picture of that on the top, that top picture right below the lamb and the cow. That's the cutaneous. The inhalation, and we see that picture of a doctor holding an x-ray, which there's problems with the pulmonary issue from inhalation of that. And then there's gastrointestinal. So it is a very old disease, okay? It's the 5th plague of the Bible. Before September of 2001, the last case of inhalation anthrax in the United States occurred in 1978. Unfortunately, in September and October of 2001, anthrax was spread through powder and letters in the mail. A number of people developed the cutaneous form, and at least 10 contracted the inhalation form, the most deadliest type. 6 of those 10 survived, probably because they were treated early with combinations of antibiotics, ciprofloxacin, or dicloxine, along with one or more antibiotics effective against anthrax. I want you to try to understand this too. When a disease first comes out, like when anthrax was first an issue, when a lot of diseases, even things like chickenpox, are first come out and they emerge and we find out about them, we don't know enough to know what works in treatment and what does not work in treatment. But the good thing is we learn, right? We take the science, we do all the science we can, we do all the data and research, and we collect that data and research, and we figure out what is the best treatment, and we share that information with other scientists and doctors and healthcare workers so that when we see this reemerge again, we have enough knowledge to better treat it. And not only better treat it to decrease people from getting sicker and dying, to have their outcomes be better, but to decrease the spread of that disease. We learn how it spreads through scientific research, and we figure out what are the best methods to decrease that spread. So we want to talk about the plague, right? The Black Plague. That was caused by Yersinia pestis, which was a gram-negative bacilli that resembles a safety pin, okay, with bipolar staining, okay? And there's a picture of that up there on the top right-hand side. So a disease of rodents, right? It transferred to humans by flea bites. The fleas got on the rats. They got disease, the fleas got on the humans, but the humans made the humans sick, right? There are 2 forms, bubonic and pneumonic. If the disease is not treated, the mortality rate is 100%. The plague is also known as the Black Death because in its later stages, if untreated, patients, the blood vessels are destroyed and the subcutaneous bleeding leads to black spots on the skin called bulbous. And you'll see a picture of someone down there, there's a fingers and then a guy down at the bottom that shows those signs. The bubonic form is a result of a flea bite, like I talked about. The patient gets a high fever, painful regional lymph nodes, and those bulbous begin to appear. The pneumatic form, that's the bubonic form, the pneumatic form is spread person to person by respiratory droplets or developed from bubonic plague into a systematic infection that affects the lungs. Epidemic outbreaks of pneumatic plagues are highly fatal if left untreated. In the Middle Ages, between 1/4 and 1/3 of the population of Europe was killed by the Black Plague. In 1891, 6 million died of the plague in India. Currently, only a dozen cases are reported in the United States each year. Chloremia is caused by a gram-negative bacilli. Human beings are usually infected by bites from infected rabbits or ticks. The disease presents with fever and flu-like symptoms. Patients present with localized or generalized enlargement of the lymph nodes or lymph vessels as a result of direct contact. Pneumatic pneumonia may be resulted from exposure to aerosols. Smallpox is caused by the variola virus. There are 2 forms. There's variola major is the most virulent strain, causing severe blisters and high fever and kills approximately half of those infected. Variola minor is the milder form with a mortality rate of less than 1%. A vaccine made from live Yersinia viruses is available and is given intermittently with a bufferated needle. After vaccination, a pustular lesion develops with induration surrounding a scab. Okay, so top two pictures are showing you, well, that very top picture is showing you the virus itself. And then the two human specimens are showing you people with the virus, and it caused a lot of death. And then from the vaccine, the thing on the little scab on your skin. The severity of the disease depends on the virus that is causing it. Smallpox has killed more people than any other disease, 300 million deaths in the 20th century. As of 1980, smallpox was declared eradicated by the vaccinations given around the world. This is why I am very much, if done correctly, in support of vaccinations. Smallpox is the only disease we have eradicated. Eradicated means you never see anybody with it. Earlier when I talked about the plague, we would see a few people every year come down with the plague in the United States. No one worldwide comes down with smallpox anymore, thank God. Okay, it has been eradicated and was eradicated by vaccinations given around the world. Vaccinations work, okay? If done correctly, they work to make sure no one not only dies from this disease, but no one gets it anymore. And again, smallpox is the only thing that has been declared eradicated, okay? And it needs to be no one getting it nowhere in the world, okay? And again, we got there by vaccination. The victory over smallpox could be in jeopardy if it were used as a weapon of bioterrorism. Some of the complications of smallpox are encephalitis and infections of pregnancy, okay? Viral hemorrhagic fever is the most lethal agent known to man. It is caused by the Ebola virus. Transmission occurs with direct contact of infectious body fluids such as blood, urine, semen, and saliva. The disease is characterized by high fever, extensive bleeding, and destruction of the internal organs. Ebola was first discovered in 1976 after two simultaneous outbreaks in Africa. The fever is spread by direct contact with infected blood of the body parts. It was spread when bodies were prepared for burial and organs were manually removed. That top picture to your right shows you the Ebola virus, and then down at the bottom to the right is areas of Africa where we had outbreaks of Ebola. Bats are the most likely reservoir, okay? Spread by infected wild animals to humans, followed by human-to-human transmission. So as you can see from this, the bats get it, they give it to other animals, and then from human contact with animals, then we get the disease. Infected persons are not contagious until the onset of symptoms. It has an acute onset, typically 8 to 10 days after exposure. Infectiousness of body fluids increases as patients become ill. Deceased persons are highly infectious. Symptoms are fever, chills, malaise, anorexia, nausea, vomiting, diarrhea, headaches, rash, chest pains, and multi-organ failure, and leads to death. Fatality rate is at 70%. This is high. This can kill you. The only good thing is it kills people so fast it is somewhat easier to contain. Botulism is a neurotoxin produced by the gram-positive rod-shaped bacteria Clostridium botulinum. It produces dry mouth, dilated pupils, and progressive muscle weakness that leads to respiratory failure and death. There are 7 types of botulism toxins, but type A is the most potent of all. A lethal dose of botulism toxin is measured in nanograms, an amount that cannot be seen. Okay, guys, so this covers Chapter 8. Like I told you, there will not be a specific test for Chapters 8 and 9, but you are responsible for that material. So once you take your test for Chapters 6 and 7, and you watch your videos for 8 and 9 and read them, I would immediately get on your review guide for your lecture final. So you guys stay safe and have a great day.